Hello, my name is Suman Rice, and I'm chair of the Society for Reproduction and Fertility. I'm delighted to welcome you to the SRF 75th Anniversary Lecture Series. This collection of talks celebrates 75 years of the Society, its rich history, and its ongoing contributions to reproductive science. The Society was founded in 1950 as the Society for the Study of Fertility, bringing together a remarkable mix of scientists and clinicians, from reproductive physiologists to endocrinologists, pioneers of livestock breeding, and experts in human fertility. Each lecture in this series highlights the diversity of research and innovation in our field, reflecting the interdisciplinary spirit that has defined the Society from the very beginning. We hope these talks give you a sense of the Society's achievements, the work of its members, and the dynamic community that continues to advance reproductive science.
Thank you very much for allowing me the opportunity to give a lecture today on the 75th anniversary. It's a privilege to be able to do this. I first did a presentation to what was then SSF in Reading in 1984. So I feel like I've come on quite a long journey with this society. In the background, there's a picture of Bob Edwards and the Nobel Prize. He was obviously an ardent member of the society, and I feel that he's watching me continually when I'm giving this type of presentation. I don't believe I have any significant conflicts of interest to declare to you and that would change the way that I would do the presentation. I'm employed by the University of Dundee. I have two editorial duties, one with human reproduction and the other with science. And I do occasional lectures, stroke workshops for companies, which may cover expenses, sometimes honorarium, accommodation, and travel. So I'm going to talk to you today about a contraceptive plan, a theory that we're trying to develop with some data. There's a clear need to improve our contraceptive portfolio, and I'll just go through one slide of that. Then I'll move on to the idea of the approach that we're using, which is looking at the human sperm cell and applying a phenotypic drug screening approach to that. And I will talk about some preliminary data that we have and also the power of this type of technology to potentially develop non-hormonal contraceptives. We are involved in other contraceptive programs and a target-based approach, etc., but I won't be talking about those today. So just to refresh our memories and really drive the point home, there is a substantial challenge in contraception. The figures actually are really quite disturbing. Even if you look, for example, at low middle income countries, there's 200 million plus people who want to avoid a pregnancy, but they're not using a modern method. Globally, about 1/3 of women discontinue hormonal methods of contraception within 1 year, and approximately 35 million women have abortions in unsafe conditions. And the statistics related to contraception go actually on, and they are quite frightening. And what that really means is that although there's been some tremendous developments in the last 50 years, our portfolio is still too restricted for contraception. And we need then to look forward and how can we develop this portfolio. So our approach in Dundee, funded by the Gates Foundation in America, is basically to target the human sperm cell. And we're using a non-hormonal approach to do that. So we're looking at different compounds, for example. And the reason why we're focusing on the human mature sperm cell is because that, of course, is the cell that actually does the work in the female reproductive tract. And we're not targeting the production of sperm, as an example. We're not targeting spermatozoa as they're driven into the testis. And the advantage of attacking the sperm cell rather than, for example, the testis, is that actually if you develop a product, non-hormonal contraception for men that affects the testis, you've got a time delay of several weeks, sometimes several months, before that contraceptive efficacy takes place. And we know that because, looking at hormonal contraceptives for men. So, and then once you've got your contraceptive effect and you want to then come off contraception, you've got to then basically come back to normal. And again, that can take several months. So we're attacking the sperm cell, when in theory, that approach can be much more rapid. The other reason is actually simplistically that we know a reasonable amount about the sperm cell, particularly the human sperm cell. It's okay. We have average knowledge about that. The target cell is readily available. So if we want to work on the cell, we have a primary cell. We don't need to culture it, for example. It's readily available, and we have a lot of knowledge about that. And of course, our approach is targeting the sperm cell in the female reproductive tract. But of course, you could flip that the other way and say targeting the male reproductive tract as well. So in general, there are two main strategies to drug discovery. You're probably most familiar with the target-based approach. So you find a target and you develop drugs to that. And that's 95% of the drugs that are in development. The problem for doing that with human sperm is simply the fact that we don't have many targets, not high-quality targets that can easily be accessed. And that is a fundamental challenge at the moment. There's a lot of work going on in trying to identify new targets, et cetera, et cetera. But at this moment in time, there are probably only two, maybe three, I would suggest two, that are potential targets in the human sperm cell. So the other approach is a phenotypic approach, where you look at your target cell, you add compounds, and then you see a change in phenotype. And quite a recent example of this approach has been with ivacaftor for CF. Of course, whatever we do, whenever the process of drug discovery, we need to understand the bar is very high. So why do we choose phenotypic screening when we're thinking of doing this approach? Well, I mentioned the lack of targets, which is a key issue. But the advantage, of course, of phenotypic screening for us is that actually it's very relevant when you've got a lot of clinical information about the assays, i.e. sperm cell, how they move, do the acrosome react, etc. But your knowledge base isn't huge. It's sufficient, but it's not enormous. And that's where phenotypic screening tends to come into its own. Also, we're looking at the final cell. We don't need to think about translating information from in vitro to in vivo because we've got the final cell that we need to target. And because human sperm cells are very readily available and we have a lot of clinical information on what's pathological, we don't need to translate this information from species to species. We are working on our final target species. But, and there's always a but, but the challenge for a phenotypic screen is actually, how do you do this? And this has occupied our work probably for the last 8 or 9 years, really. And this slide just summarizes sort of 5 years of work, or 5 years of thought process. And one of the key things that we learned quite early on is drug discovery is a professionalized system. You can make many, many mistakes if you're not collaborating with professionals in this area. And so that's where our collaboration is with drug discovery by the Gates Foundation in Dundee. The other thing was that we spent a lot of time trying to think about it. Assessing movement in sperm in high throughput was deemed to be very difficult at the time. So we always tried to look for a surrogate, looking at calcium measurements, etc. But those don't really work. What we really want to know is the target cell and its pure function, not a surrogate. So initial experiments really tried to sort of get a system where we could add many compounds, and by that initially we're talking about 3 or 4 a day, and then seeing the effect on the phenotype. But we needed to scale up, and we were having lots of challenges doing that. And we needed a rapid system. And basically we needed a system that's going to examine thousands of compounds a day, not 2 or 3 or 10 at most. So fortuitously, I saw an advert in the back of Nature, actually, for the Grand Challenges for the Gates Foundation 2017, a call for a phenotypic high throughput system. So collaborating with Paul Wyatt in Drug Discovery Unit at the time, we wrote a very brief proposal. It was literally 1 A4 page to the Gates Foundation. And we were successful in getting that. And we got Franz Gruber, who transformed the way we were trying to do this project, completely transformed it. So he was basically working in the National Phenotypic Screening Center, trying to see if we could adapt robots to looking at sperm motility at the same time as adding compounds and seeing a phenotype. And sort of literally within about 3 or 4 months, Franz had cracked the system. It was just amazing what he'd managed to achieve. Incredible, talented individual. And what he got was basically you put the sperm cells, prepare them, put them into the wells, 384 wells, and then you put them through the automated phenotypic screening. And he devised a system to look at sperm motility, and he did all the controls of looking at positive and negative, etc. And what he showed was that actually you could screen a significant number of compounds in one day. So we could probably put 4 or 5 plates in a day when we were looking at sperm motility. And this was a revolutionary development actually for us. And now what that means is you can get a library of compounds. So you might be interested in a library for G proteins, a library for ion channels, whatever your main interest is, or you want a repurposing library. And of course, that's how they developed Viagra and sildenafil. But so we looked at a repurposing library to start with as our first experiment, really. So this is about maybe 12,000 compounds. And then you can see basically on the graph, enhancement of motility, which of course for contraception you're not interested in, but for infertility, but that's a different matter. We're interested in reducing sperm motility compared to the controls. So we found a number of compounds that reduce motility compared to the controls. And this is screening a large number of compounds in an automated, reproducible, reliable manner. So we were incredibly excited to get in this data. And not only were we just really excited by the power of the data, we were excited by the sort of the results that were coming up, which were consistent. You know, for example, we were seeing compounds that were toxic to sperm coming out as toxic. And that was obviously very, very encouraging. We were learning a lot about the sperm cell as well, you know, adding, for example, kinase inhibitors, what do kinase inhibitors do to sperm motility, etc. So we were getting some tremendous information and learning simple things such as, you know, antifungals have quite a powerful effect on sperm. The second thing that was really quite interesting was that actually sperm are pretty robust compared to other cells. When we're trying to drug discovery people, they say, well, actually, you know, they're pretty hardy, these chaps. And that was something that an important thing that we were learning. But scientifically, what was really important was that we could then, if we found an interesting compound, we could do then modifications to it. Well, I say we, I mean the chemists, and then add then 50 or 100 different these modifications to the sperm cells. Same sample, which is, of course, improving our reproducibility, reliability, and then we could see an effect. So it's a very powerful, constant feedback loop to try and look at these compounds. So it all sounds incredibly interesting and exciting. But, you know, drug discovery is a lot of failure, you know. So we've screened many, many compounds, and this just shows an example of this. And Anthony, if you're interested, has just recently published a paper putting together a screen of about 88,000 compounds. So we picked them from various libraries. We picked them from standard libraries, Select Chem, for example, where there's sort of quite a lot of information about known compounds. We've done diversity sets, which actually we don't know we know what the compounds are, but of course they're not yet being screened in other cells. And the target, for example, is unknown. And you could screen ion channel libraries, which we've also done as an example. But of course, then when you look at that and you look at the data and say, okay, well, how many of these have really hit sperm motility significantly that we think would be clinically relevant, but are not toxic? Of course, that's no good being toxic to other cells. Then actually, as you start to go down this cascade, this triangle really, really starts to pin at the bottom. So of 88,000 compounds, we found only 9 series which were of particular interest for us to sort of look at in any further detail. So why only 9 series? It turns out that actually we can stop sperm by being very toxic chemicals. That's not much help because they're going to be toxic to other cells. We were going for quite a strong effect on progressive motility, so we had quite a high bar. So many would have a tickling effect on sperm progressive motility, but that wasn't clinically enough. We need to make sure there's no somatic toxicity. We need to test against other key somatic events, and of course, one of these is using hERG channels. Sometimes we find a compound which fulfilled a number of criteria, but actually there was limited chemical space around the compound that you could basically, I would say, play with, but that's not a technical term. You could modify to then improve its potency or reduce its toxicity, etc., etc. You know, anything can go wrong, it goes wrong. So you generate huge data, but you know you end up with a lot of dropout. And in fact, of the 9 series, we're only following 1 series at the moment, but we have been following that series for 3 years, and that's a very profound effect on progressive motility. And we've got a lot of information related to the safety of that compound. Interestingly, when we were doing this, and I was blown away by Franz's ability to develop these assays, it suddenly occurred to us that actually we're focusing on sperm motility because it's clinically relevant, but actually we could look at other things. You know, scientifically, if you're interested, for example, in membrane potential of sperm cells, then as long as you've got a good stain and a good dye, basically, that's robust, we can test it in this system. If you're interested in acrosome reaction, then you've got a good assay for acrosome reaction, PNA, PSA, or CD46 or whatever. We can then test it in the system, etc., etc. And actually, this really opened up a new field, a way of thinking about how we could look at the biology of the cell. Now, and I won't go into a lot of detail about that because it's not directly related to contraception. But when we were doing this, we then thought, well, we're looking at the basic sperm motility, but of course, sperm in a female reproductive tract are in a capacitated state, or undergoing a capacitating condition, rather. Therefore, we should focus on that as well, because if we're targeting the female tract. So basically, Franz developed a capacitation assay, and as an output of this, he looked at hyperactivation. So you put the cells in capacitating media to cook them up a bit, basically, and then you see whether the cells can sort of hyperactivate as a marker of capacitation. And then you can incubate them with compounds and then see if the compounds affect capacitation, hyperactivation. But the advantage also of this is you can look at progressive motility as well. So that's an advantage of this assay. You can look at progressive motility, total motility, hyperactivation. It allows you a lot more information from basically the same assay, looking at sperm motility. And of course, we've set this system up where you would assess capacitation in the presence of the compound. But of course, you could put the compound in first to block the process, or you could put the compound in at the very end and see if it blocks a terminal event. So the flexibility of the system is something that actually I hadn't appreciated in the beginning. And now that is becoming very powerful for us. So if I give an example of, say, the capacitation data, so this is unpublished at the moment. Again, going through a series of libraries that we particularly find of interest, standard libraries and libraries that say, focus ion channels, because we're quite keen on what ion channels do for, say, hyperactivation, capacitation. And what we can see from these, and this is screen about, at the moment, this is 10,000, we can see some decreased hyperactivation and obviously decreased general motility. Some just affect decrease in hyperactivation. They have no effect on progressive motility, which is I think particularly interesting. And as a side issue for the aficionados, generally they don't affect cat sperm, which is quite surprising. I was quite surprised. But anyway, so it looks like it's a separate assay to cat sperm, which is a very positive actually. But of course we can see compounds that increase hyperactivation. And actually, first of all, I thought, well, they wouldn't be a contraceptive, but maybe they are. Maybe we can overcook the cells. That might be an approach that we could use. Anyway, so we're I keep saying playing that. We're moving with this assay forward and trying to sort of then look at a number of these series. So we developed so far 2 series, which are particularly interesting in this arena. And for those of interest, we use a SAC inhibitor as a control. So with any system, I'm obviously very excited about this system, and I'm an enthusiast, shall we say. But there are challenges with any system. Phenotypic screening is a balance. It's a compromise between preparing a sample and getting the most detailed kinematics versus high throughput. So, for example, this is 45 frames that we get. We're not looking at huge detail for the kinematics of the cell. We're pooling samples together because we need large numbers of cells. Again, that's there's advantages and disadvantages to that. When we're looking at some of the other assays, it's less clear to us what the clinical cutoffs are. Obviously, 0 is always a good one. They don't acrosome react, no motility, but generally compounds don't do that. So how much wiggle room do we have there? That's an important concept. And of course, just because we find something that inhibits hyperactivation and get all the chemicals detail of it, and it's all very exciting, that's actually just a start. We need to do many other experiments to work out what's happening with this. And although I say we're using the final cell because we are the sperm cell, and in fact, this case, we're capacitating, so it's about as final as we can get it, it still is out of context because actually sperm work in the female tract, and we'd be targeting in the female tract. So what they do in the female tract in the humans, we know is 0. So we are doing a lot of guessing about timings, about interactions, etc. So it's as close as we can get it, but it still isn't the final phenotype. And of course, anybody who's involved in drug discovery will tell you about phenotypic screening. The Achilles of this is mechanism of action and target. So when we find an interesting compound, we have to find a target to that. Now, there's complicated proteomic type ways of doing that, way out of my expertise, but luckily we have collaborators who can do that. But you do need to then progress to a target. So just to start to summarize now, it's very exciting being involved in this area. I've moved from infertility to contraception, so to speak, so it's been a journey for me personally. But it is a very long road, drug discovery, and I probably had underappreciated that when I was getting involved in it. But having an assay like this and be able to test these compounds and then do safety is a massive plus, and that really does give us a leap of faith in the approach that we're using. But it's a very, very high safety bar. You know, we need to do much reproductive toxicity testing. We need to know if the compound will get to the site of action. So how will it get to the female tract, in what concentration? Sperm survive for 7 days in the female tract. How do we interrupt them? There's lots of questions we need to ask. And, you know, we've got to answer those questions. But we've got the basic system and the collaboration with drug discovery sorted. And that is a massive leap in the development of non-hormonal contraceptives. At least targeting the sperm cell. So hopefully I've given you a bit of a flavor of the approach we're using in Dundee, which is a phenotypic approach using the sperm cell. Hopefully I've given you some feel for the urgent need to change and develop the contraceptive portfolio. And while it is a long road, and there's no doubt about that, I think we've made substantial progress into really knocking on the door to development of non-hormonal contraceptives. Now, it is impossible in this work. I just have the privilege of giving a lecture on it, but the people behind the work are really the people who deserve the credit, not me. And that involves a collaboration between Nine Wells Hospital Reproductive Medicine Group and the Drug Discovery Group run by Ian Gilbert. We also have collaborators in Virginia, in the United States, the Modo Action Group who work with Ian, and NPSC, Jason and the team who also work with Ian and the team. So this is very much a team approach to doing that. I've just got the privilege of actually saying what we're doing. And on that, thank you very much for your time. And any questions, just feel free to give me an email.